All right, hello everybody. It's nice to see you after a pause. I can see that I'm scaring some of you, that you have to sit at the very back, but that's fine. I'll try to speak up so that you can hear me. So you are now in a course which deals with signaling, with the different mechanisms of receptor action and signaling cascades and whatever. And today we are starting kind of a mini series, mini block, where we will be talking about neurobiochemistry and signaling in the nervous system. So we will really focus on the different signaling molecules in the nervous system, the different principles behind it, all the different receptors and what they do. And towards the end of this mini block, we will also talk about the implications of these principles into pharmacology, neuropharmacology, and psychopharmacology, and how we can influence the signaling mostly in the central nervous system, but also in the peripheral nervous system. So that's kind of the plan for the next, 5 lectures or something like that. Today's lecture, which is the first one in this kind of neurobiochemistry mini course, is going to focus on the basic modalities, the basic ways how information is transmitted in the nervous system. Okay, so we'll not be talking so much about individual signaling molecules. We'll be talking about some molecules, but mostly it is really to give you an overview of what kind of basic principles of information transmission we have, so that we can then later on plug in the specifics and we can see how things work in the specific cases of specific structures and specific molecules. So before we start talking about what those principles are for information transfer, let's just briefly review the composition of the nervous system. And let's focus on the central nervous system, because the peripheral nervous system has its specifics, but we'll mostly be thinking we'll mostly be thinking about the central nervous system, so about the brain and the spinal cord. So what do we find in these organs?
Nerve cells and glia.
Excellent. So we find nerve cells, glial cells, and maybe some other types of support cells. Very good. Now, what is the proportion? What are the proportions of these cells? Are they about the same? The number is about the same, or is some of them more abundant? Okay. Okay, so in many textbooks, and kind of the tradition says that there are many more glial cells than nerve cells. Latest research is showing that this does not seem to be true. Okay, there are actually fewer glial cells than there are nerve cells. They are about the same, but there seem to be fewer glial cells. There are good reasons for this original kind of myth of 10 times glial cells and whatever. And I was, just a few years ago, when I was standing here, I was saying this myth. But latest research where they use more modern techniques, how to count cells, because you can imagine that counting cells in the brain, where there are a lot of cells and they are really, right next to each other, it's very difficult. And the cells are different sizes, etc., so it's not easy. But the latest research is saying that approximately, if we take the brain or the central nervous system as a whole, the overall ratio, which can be slightly different in different structures, is about 5 parts of nerve cells to 3 parts of glial cells to 1 part of endothelial cells, because there are a lot of endothelial cells because you have all the blood vessels that need to supply the brain. So there are actually a lot of endothelial cells there. So this is an approximate ratio that currently is being accepted as the average over the whole central nervous system. Again, some structures may differ. So, even I used to be saying 10 times more glial cells, etc. It doesn't seem to be actually correct. So these are the approximate ratios. So there seem to be actually a bit more nerve cells than the supporting cells. We will mostly be talking about the nerve cells and what they do, but we'll also at times mention the glial cells, and most importantly astrocytes or astroglia, which play a super important role in the energy metabolism of the brain, but also in metabolizing neurotransmitters. And to some extent, they also transmit signals between different parts of the brain. Actually, astrocytes are connected by gap junctions to each other. So, could you close the door, please? Yeah. So they can actually transmit excitation. They're not they can't be excited the same way as neurons, so they can't form action potentials, and we'll talk about action potentials today, what they are. So they can't do that, but they can still transfer some information by changing ion currents, etc., etc. So we will mention astrocytes especially, and we'll also mention a little bit about the endothelial cells, because they are the crucial part of the blood-brain barrier, together with astrocytes, and which controls what can get into the brain or into the central nervous system, what cannot get there, and what will be exported, etc. So mostly it's going to be nerve cells, but we'll be talking about the supporting cells as well. Good. So let's now talk about the two main modalities, the two main ways of how information is transmitted in the central nervous system. The first one is electrical, by means of basically electromagnetic pulses, changes in the electromagnetic field, okay, which is mediated by changes in the membrane potential. Okay, so we'll be talking about that quite a bit. Now, this electrical way of transmitting signals is especially used within one nerve cell. Okay, so one nerve cell will be using mostly this electric way of transmitting signal along the length of its axon, as we'll see in a second. But we now know that there is actually electrical signaling even between different nerve cells by means of what is usually called electrical synapses, but really, so I'm gonna give you this terminology, electrical synapses, even though I'm not sure it's a really good thing. Basically, what we now know is that between many nerve cells, there are gap junctions, and you've heard about gap junctions because they allow the flow of ions between different cells. So these gap junctions work as so-called electrical synapses, and they basically, without the need for any signaling molecules, they can transmit excitation between nerve cells. Now, this is a relatively new finding. So just a few years ago, we would say, well, yes, there are electrical synapses maybe in the retina, but maybe we don't know what they're really doing. Now we know that there are actually gap junctions all over the central nervous system. And one of the functions, probably not the only one, but one of the functions of these gap junctions is to synchronize neurons to work together. Many of you have heard about EEG, so about the method where we basically follow the electrical signals in the brain as a whole. And you know that these waves basically, they oscillate. So, and these oscillations are in part formed by this synchronization of brain cells, which tend to work in unison, basically. And this is probably, at least in part, mediated by these gap junctions, okay? So-called electrical synapses. So again, just a few years ago, I would be saying, okay, electrical signaling is just without one cell, just within one cell, but between cells, not so much. Now we know that in the central nervous system, actually, this electrical signaling is probably very, very important, okay? You know that gap junctions are formed by some proteins, connexins, and these connexins, their opening and closing can actually be regulated. So it's not like they're just holes between cells where anything flows, but the cells themselves can actually regulate whether they open them or close them. So this actually, in neuroscience research, this opens a new window into signaling between cells where they can regulate what they allow to go through, and we know very, very little about it at this point. But so I just want to tell you that gap junctions are probably very important. But when we talk about electrical signaling, we'll mostly be focusing on the single axon, the single cell, because we understand those processes quite well. The other modality, apart from electrical signaling, is chemical signaling. Basically, secreting signaling molecules and allowing them to bind to receptors, and those receptors then start some signaling cascades in the next cell. And this is the principle behind normal synapses or chemical synapses. Yeah, so that's a normal synapse or chemical synapse where one neuron secretes stuff into the extracellular space, and there are receptors on the postsynaptic neuron which are activated or deactivated or whatever, which react to these molecules in the same way as immune cells will react to cytokines or whatever, okay? So those two modalities, today we will discuss the principles behind them, how these things actually work. And then in the coming lectures, we'll start talking about specific molecules and specific receptors, et cetera, et cetera. So that's the plan for today and also for for the mini course. So, talking about the electrical signaling, as I said, it's basically signaling by changes in the electromagnetic field. Okay, I know this sounds very abstract, and what does it mean? But really, it is mediated or is caused by changes in the potential, in the electrical potential on the plasma membrane. So we're changing the polarity or the voltage on the plasma membrane, and as this change moves through space, it's basically changes in the electromagnetic field, you know, in a similar way as a radio or something is sending signals through the electromagnetic field. It's a similar principle behind it. Now, how does it work? If we want to change the polarity or the voltage on the membrane, we first need to have a voltage on the membrane, right? So most neurons, or almost all normally functioning neurons, will have a certain potential difference across their plasma membrane, which is called the resting potential. So the cell is not doing anything, okay? But even when it's not doing, I mean, it's obviously doing a lot of stuff, right? But it's not being activated, it's not being excited. It will have a certain resting potential. Now, the usual resting potential of a neuron is approximately minus 70 millivolts. Again, this may change between different structures of the brain, different types of neurons, but approximately minus 70 is a usual value, okay? Let's say 60 to 80 or something like that. Now, when we say it's minus 70, that means that it is negative inside. This is by convention. We could easily call it plus 70. It doesn't really matter, right? But the convention is that we're taking the negative inside, so it is negative inside. It's minus 70, right? How is this resting potential achieved? How come that these cells actually have a resting potential? What is it caused by? By ions, by proteins, correct. Both of them play a very important role. So let's go into a little bit more detail which ions and what about them, and which proteins and what about them actually give these neurons a resting potential of about minus 70. Sodium and potassium. What about them? You're right, but what about them? Well, let's stay with the ions first, and then we'll talk about the channels, okay? They both are needed, okay? So you say sodium and potassium ions, correct, but the concentration difference, excellent. So what is the concentration difference? So if this is the plasma membrane, this is the outside, and this is the inside. Just talking about sodium and potassium, okay, what are the differences? Where there is more of those or less?
Potassium is more inside of the cell and sodium is more outside.
Correct. So we have much more potassium inside, much more sodium outside, right? And vice versa, okay? So we do have a concentration difference also in other ions, but most, now let's talk about sodium and potassium, right? Now, this on its own will not create a difference in electrical potentials because these concentrations of potassium and sodium, they are balanced by anions, which are there as well, right? So in the beginning, there is no electrical difference between them. We have the same number of positively charged and negatively charged ions inside, and the same number of positively and negatively charged ions outside. This is quite important to understand, okay? So before we get any membrane potential, we do have differences in concentrations of these specific cations, but we also have differences in anions concentration and other ions. So overall, they are electroneutral. There is no membrane potential just caused by these concentrations, by these concentration differences. Is this clear? Because I think this is often something that students might get confused about. Is it clear that we have a difference in sodium and potassium concentrations, but if we add all the ions, all the positively and negatively charged ions inside and outside, we get exactly the same number. So there is no electrical difference to start with.
What do you mean to start with? Like in which stage?
Before any membrane potential develops.
But there's always room.
Well, yes, if you look at a functioning body, there is a membrane potential because otherwise it wouldn't work, okay? But this is a model to explain how we can keep it, how we can replenish it, and how we can keep the membrane potential, okay? So let us start now with just differences in these two concentrations, but overall it is electroneutral. You're right, in a living organism we will never see this because it already starts with the membrane potential, yeah? So So it's a model. Yep.
So you're saying that if we have 100% of like the molecules in one side, it will be equal?
Yes. So all the potassium, sodium, chloride, proteins, bicarbonate,
Is zero.
if you sum it all together, it's going to be 0. And here it's going to be 0 as well. Yes. Okay? To start with in our model. Okay? So we do have differences in ion concentrations, but that's not enough to give us the membrane potential. What do we need for that? Well, sodium-potassium pump will influence the membrane potential, that is true, but it's not the most important bit to actually start it. We need something else.
The leakage of potassium?
Correct. So in most resting cells, there will be in the membrane potassium channels which are open all the time. Okay? So the membrane is impermeable to all other ions. This is a model, it's not true. Okay? In real cells we have lots of different permeabilities, but this is the easiest way to understand it. So we will only have potassium channels which are open in the membrane. So the membrane is only permeable to potassium. What will happen?
Outflux.
Correct. So because there's a lot more potassium inside, the potassium will start flowing out, right? Because there's a concentration gradient, so just by diffusion, there's going to be a net outflow of potassium ions from the cell, right? Now, what will that cause, what will that do to the potential difference, to the electrical potential difference? So we started with 0, we started with the same on both sides, but correct. So positive charges are flowing out. That means that the inside is going to get more and more negative, right? And the outside is going to get more and more positive because positive charges are flowing out. None of the other charges can flow out or in. So this is the only thing that is happening, right? Good. Now, this outflow of potassium will actually stop at some point. And it will stop way before the concentrations become the same. It will actually stop, and we already talked about it in a lecture in the first course, it will actually stop at a potential approximately 90 millivolts, minus 90 millivolts. It will stop there. Why will it stop there? Why will it not just keep flowing out?
Because it's also attracted inside.
Correct, because we have now electrical field which is pulling these ions back in, and the two forces, they're not really forces, will be equal at some point. And this is the equilibrium potential for potassium. So potassium will stop flowing out here because the electrical force is pulling them back in, right? Now, I said that in real neurons, we don't really see minus 90. It's approximately minus 70. And the reason is that this model that I just showed you is really simplified because we will have other channels. We have NAK ATPase, and they're all working together. So usually we are about minus 70. Okay? But this simplified model tells you kind of 90% of what causes the membrane potential. Okay? So it's not really NAK ATPase. Okay? Even though NAK ATPase is electrogenic, it will increase this potential or make it more negative, but it's not really the main cause. The main cause is this leakage of potassium ions. Okay? Simplification, but I think a useful simplification. All right, so that's our resting potential. Now, let us have a look at one individual neuron in the brain. And we'll take a multipolar neuron because there are many of those, but as you know, there are different morphologies of neurons, so they don't have to be multipolar. So we are going to have a body with some projections, right? Here is the body with the nucleus, and here we get one long projection. So what are the parts? What are these things called? Dendrites, yeah, very thin projections. There can be many of them, or it can be just one of them, depending on which neuron we have. They may have some dendritic spines, etc. There are all sorts of different structures there. This is called the soma, or the body of the cell, right? Why is it important? Yeah, so we have the nucleus there, and we have all the protein production there. So all the proteins for the cell, even for the axon, are actually being produced in the body, in the nerve body. This is very important, and we'll see it towards the end of the lecture, how we can exploit the fact that no proteins are synthesized here. They're all synthesized here, okay? So yeah, the nerve cell body, lots is happening. And the last bit is this thing, and that's called the axon, right? Each neuron has how many axons? One. Excellent, okay? So there's just one axon. That's how it works. Now, in order to understand how the signal is transmitted, the electrical signal is transmitted in each individual nerve cell, we have to look at the difference in membrane composition and difference in conduction abilities of these different parts of the cell. Okay, this sounds abstract, but I'll show you very specific examples. So the dendrites are there to really gather signals from other nerve cells, right? So basically we have other axons coming to the dendrites, forming synapses with them and releasing some neurotransmitters into the synaptic cleft. Okay, this is something that we'll cover in more detail in a minute, okay? But this is what happens. So some kind of electrical signal comes here. Here it's converted to chemical signal. So some stuff is being released. And on the dendrite, we have some receptors which will react to these neurotransmitters and will do something. We'll open some ion channels or start some signaling cascade or what have you. Something happens, right? Now, one of the main things that we are interested in happening here in the dendrite is what happens to the membrane potential. Okay? There may be other things happening, but let's leave those aside. But we will be interested in how this signaling from all those different axons which come from other neurons, how it affects the membrane potential. And there are basically two possibilities. So either it can make the membrane potential more positive, and we call that depolarization, absolutely. Or it can make it more negative, and we call it hyperpolarization, okay? So depending on the type of receptors and the type of neurotransmitter, the membrane can be either hyperpolarized, become more negative, or it can be depolarized, become more positive, right? Now, all these changes in membrane potential will then start diffusing along the membrane towards the body, or rather, they will actually start diffusing in all directions, but it's just diffusion, right? So we allowed some ions to go in or to go out, depending on what goes on. And the change in the potential and in the ion composition will just diffuse along the membrane. Now, this is quite crucial because in the dendrites, this way of moving the charges and the change in membrane potential by diffusion means that the signal is becoming weaker the further it needs to go. So here it's the strongest, right? The biggest change in membrane potential. But since it is just by diffusion, it will become weaker and weaker and weaker and just moves along the membrane and becomes weaker and weaker, right? So these kind of potentials and spreading potentials are called dendritic potentials. Okay, they are just diffusion of those ions, changes in membrane potential, but it is weakening as the further it goes, it is weakening, right? So they're called dendritic or dendrite potentials. But a big change, so this all happens along the body and everywhere, okay, there's just this diffusion. A massive change in the type of propagation of the signal occurs in the very beginning of the axon, which is called the initial segment. It's called the initial segment. And it is the first part of the axon. And there, this slow diffusion of hyperpolarization and depolarization basically is all summed up, okay? So all the changes in potentials which are coming from the dendrites are all summed up. So all the hyperpolarizations are basically subtracted and the depolarizations are added up. So we get a final value of the membrane potential, okay? Some value of the membrane potential here. And if the value of the membrane potential is more positive than some threshold value, a new type of potential develops, which is called the action potential. So I'll say that again. We have these dendritic potentials, which are just moving along by diffusion. Here, we take them all together, we sum them all together. And if the resulting potential is more positive than some threshold value, let's say minus 50 millivolts or something, it will be slightly different for different neurons. Okay, so just take it as a ballpark value. So if it is more positive than about minus 50 millivolts, action potential starts. Now, what is an action potential? An action potential is basically just a membrane depolarization which moves along the membrane. So that's quite similar to our dendritic potentials. But here the crucial difference is that in an action potential, the depolarization at every point of the membrane is amplified to 100%. It's not getting any weaker. It just, at every point, it is 100% intensity. So even though some of the axons in our body can be extremely long, right? The axons that are moving my small toe, they start somewhere here in the alpha motor neuron in the spinal cord, and they have to go all the way there. It's a really, really long axon, right? So if we are just relying on diffusion, the signals would have to be enormously strong just to get there, okay? But we have this action potential thing where this even relatively small depolarization is amplified at every point, is amplified to 100%. So no matter how long the axon is, we still get 100% signal at the end. I'll show you in a second how that works, okay? But this is the crucial difference between dendritic potentials, which are just diffusing and weakening, and action potential, which is not weakening. It's 100% all the way.
What do you mean 100%?
Intensity. Okay, so the intensity or the level of depolarization. So imagine that here some neurotransmitter, some depolarizing neurotransmitter was released, and this part of the membrane was depolarized from minus 70 to minus 60, or whatever, minus 30, it doesn't matter, okay? Now, so here we have minus 30. But since it is moving by diffusion, here it's going to be minus 35, and here it's going to be minus 40, and it's going to go down and just getting weaker and weaker and weaker.
Diffusion, you mean like molecules go out?
Correct. The ions are just diffusing along the membrane. Okay. So let's say that some sodium ions have flowed in here, and they're just diffusing along. So they get, the concentration gets lower and lower and lower and lower. So in the end, maybe it's never gonna, the signal is never gonna get here because it's just too weak. Okay. In the action potential, we're gonna have depolarization to, let's say, 0 millivolts, and it's gonna be 0, 0, 0, 0. It's always 100% strong, basically, 100% intense. Huh?
In the axon.
In the axon, correct. Yes. There is no action potential in dendrites, okay? And I'll give you a reason for that in a second, okay? No action potential in dendrites. Good. So are we all on the same page with this difference between dendrite potentials and action? Yeah?
I still don't get why doesn't it change?
I'll explain it. I will give you the details right now, okay? So the reason why we get action potentials here in the axon, but not in the dendrites, is one type of ion channels. And these ion channels are called voltage-dependent sodium channels. The usual abbreviation is NAV for voltage. And they are voltage-dependent or voltage-gated ion channels for sodium. Now, what does it mean that they're voltage-gated? What does it mean? Correct. The thing that opens and closes them are changes in membrane potential. That's what voltage-gated or voltage-dependent means, right? And it is the concentration, the number of sodium or voltage-dependent sodium channels that determines whether we're going to have action potential or whether we're just going to have some dendritic potentials. Because the concentration of sodium, of voltage-gated sodium channels in the dendrites is very small. There are a few, but there aren't a lot. In the axon, we have a high concentration of voltage-dependent sodium channels, and they allow the propagation of the action potential. How does it work? So, this is a voltage-gated sodium channel, right? And it is, in the beginning, it is closed because the membrane potential is minus 70. Okay, so this is our resting axon, let's put it that way, okay? Now, when the polarity of the membrane changes and we get to some threshold value for the ion channels, let's say minus 50, it doesn't have to be exactly that, but let's say minus 50, this channel opens up and allows sodium to go in, right? Sodium will want to go in because there's much more sodium outside than inside. So sodium ions will want to flow in. Now, what will this inflow of sodium do to the membrane potential? It will make it more positive. So it will depolarize the membrane even more, right? Now imagine that next to it, there is another voltage-gated sodium channel, which in the beginning is closed, but the inflow of sodium will depolarize the membrane around it, so this one will open as well and allow sodium, which will cause further depolarization of the membrane. And this is action potential. So it just moves along the membrane, and since there are lots of sodium, voltage-gated sodium channels, the depolarization is always 100% because we get complete depolarization, lots of sodium flows in, and then it moves along the membrane, new channels open, new channels open, new channels open, and it just goes on, and there's always 100% intensity. Okay, so this is not just by diffusing the ion, just by letting the ions kind of diffuse and change the potential, which happens in the dendrites. Here, at every point of the membrane, we open new ion channels which depolarize the membrane further. And this is the principle behind the action potential. Does it make sense? Yeah.
If this is the case, why sodium do not accumulate in the neuron, and then they can't have another action potential anymore?
Yes, I will answer that question in a second, okay? Because we're just getting there, okay? So this is just how to make sure that the depolarization never peters out, okay? We always get more and more because we're opening more and more channels, so we get always 100% depolarization.
Should we think about action potential as an electrical wave?
Yes, it is just basically the movement of the depolarization along the membrane. That's exactly what action potential is.
It's ions, but we look at it as like electrical wave.
Yeah, because the ions are really flowing into the cell, that's correct. But we are really interested in kind of the electrical change which is moving along, okay? So it really is about electromagnetic field and how it changes. But I understand that a lot of you have not really done a lot of physics, so I think it's a little bit easier maybe to think in terms of channels and stuff like that. But yes, in fact, it is a disturbance in electromagnetic field which moves along the axon. That's the real information thing, okay? But the molecular mechanism behind it is the ion channels and ions flowing in. Yeah? Good. So we have this perfect, nice 100% depolarization moving along the axon. But at some point, we need to stop it, right? Otherwise, we would only be able to send one action potential in the axon, and that would be it, right? We would just be, and we would just end up with all the ion channels open. And how do we stop it? Because remember, these are voltage-gated ion channels. So we can't really close them before the membrane returns to the original potential, right? Because it's the potential that opens them and closes them, right? So if everything is depolarized, basically there's no way to close them again, okay? Everything will just keep being depolarized, and all the sodium will flow in, and the axon will just burst open because osmotic pressure and et cetera, okay? So we need to have some mechanism to stop this. And the mechanism is actually built into the ion channels themselves, because inside the ion channel there is another gate here which automatically closes after about 1 or 2 milliseconds. So the membrane potential changes, the membrane depolarizes, this gate opens, sodium can flow in, and about 1 or 2 milliseconds after that, this other gate automatically closes. And the channel becomes inactivated. It's not closed, it's still open, but it's inactivated because this thing is blocking the flow of ions into the cell. Okay, so the action potential is moving along the axon, and behind the actively newly opened channels, we actually have channels which are still open, but they are inactivated, so no sodium is flowing in. Okay, and this stops the flow of sodium so that we can repolarize the membrane and return everything back to the original state so that we can send another action potential back in. How do we repolarize the membrane? Well, the main way of repolarizing the membrane is by opening more potassium channels. So, in fact, these potassium channels are also voltage-dependent potassium channels. So they open when the membrane depolarizes. They open almost exactly at the same time as the sodium channels. There's a little bit of delay, not much, but a little bit of delay. And these potassium channels, which open right after them, will allow potassium to flow out. And since this channel, the sodium channel, is already blocked, it's already inactivated, so no sodium is flowing in, it's going to be just the potassium flowing out, which will restore the potential back to the resting potential. Okay? This, the repolarization of the membrane, the return to the resting potential, then also returns these voltage-gated channels back to the original state where the inactivation gate is open and the normal gate is closed. I will redraw that so that you can see the whole cycle, because I can see that this can be a little bit confusing. So at a resting potential, we start with a channel which looks, like this. It's closed, and the inactivation gate is open. This is resting potential. Now, when there's depolarization, the closing gate opens, and sodium can flow in. And the inactivation gate is still open for 1 or 2 milliseconds. And then automatically, after 1 or 2 milliseconds, the inactivation gate closes and the channel becomes inactivated. And then we have to repolarize the membrane back to the normal resting potential, and the whole channel returns back to normal.
And the repolarization just happens because potassium always leaves the state.
Yes. In fact, we actually open even more potassium channels than we have at the resting state, okay? Because they are voltage-dependent potassium channels. So when the membrane depolarizes, a lot of new potassium channels open. And once we close the sodium channels, it's the potassium, the alpha potassium, that will return the potential back to normal, right? Now, you might ask, well, but we still have lots of sodium that flowed in and some potassium that flowed out. We have to return that back to normal. And this is the role for NAK ATPase. So this is when NAK ATPase actually comes in to return the numbers of ions back to the original state. Okay? So this is what NAK ATPase actually is important for. However, and this is quite fascinating, the number of ions that flows in through one action potential is very small. It's very, very small. It's like 10,000 ions or something like that. It's tiny. Okay? So compared to the volume of the axon doesn't even know that the action potential happened. The change in concentration is very, very, very small. It's still there, so if you just kept, if you blocked NAK ATPase and you just kept sending action potentials, at some point it would stop working, okay? But not after one action potential. The change is tiny, okay? So we need NAK ATPase, but we need it kind of in the long run, okay? Not just for one action potential. As I say, the nerve cell just wouldn't even notice that it was there. The changes are very, very small. Good. Now, this mechanism of inactivation is absolutely crucial, not just for the repolarization to get everything back to normal, but it gives us another important property of how these electrical signals are sent through the axon, and that is that the action potential always runs only in one direction. Okay? So if it starts here in the initial segment, it will only run this way. Because you can imagine that if you just, if you had depolarization here, you would think, well, what stops it from running in both directions? Because we have depolarization, so theoretically these ion channels could open as well, and these ion channels could open as well, and you could actually have action potentials running there and back again, and the axon would just be all the time depolarized. But this inactivation property causes the fact that when we have the action potential is here somewhere, behind it, there's going to be an area of the axon where all the sodium channels are inactivated, right? Just by means of this time delay. So here we have depolarized ion channels, and behind it, they are all inactivated. So the action potential can never start running back because all the channels are inactivated. They cannot open until the membrane repolarizes. So this inactivation makes sure that the propagation of the action potential is always in just one direction and it doesn't go all the time there and back, because that would be useless. Okay, so this is all done by this inactivation mechanism. Yeah.
So can we say always the signal should start from the beginning of axon?
Yes.
Otherwise, it may go.
Correct. Yeah, so if you do it like artificial in the lab, if you just put some electrodes here and put some, depolarize it electrically or something, and you do it here, it will run in both directions, absolutely. Yeah, but physiologically, this doesn't really happen. So usually the action potential starts here. But yes, you're right. You could do it artificially. Yeah. But even in that case, it would not just keep running this way and that way. It would just run in both directions and stop. Right. All right. So this is a fairly important bit of neurophysiology and neurochemistry, and you will use this model even in later years as well. So this is quite important that you understand these basics. Okay. So if not, keep asking questions. But if you, think about questions that you might ask, but I will ask you a question. So we have this voltage-gated ion channel, right? It responds to changes in voltage in the membrane potential. How does that work? How would you make an ion channel, a bit of protein, that can respond to changes in voltage? Just think about it for a bit, okay? Don't think the first thing that you think about, okay? Think about it, okay, how would you make a protein that changes its conformation, changes its structure, based on changes in voltage?
By neurotransmitters, maybe? By neurotransmitters?
Yeah, but here it's not binding any neurotransmitters. It's only responding to voltage, right? So how we have to actually build in the stuff into the protein, because the only thing that changes is the voltage, right? Of course, you could have it bind stuff, but that's not what's happening here. It's just about the voltage.
Positive and negative amino acids.
Correct. And how would you do that? So yeah, it's something about charged amino acid side chains. That's going to be the crucial bit. And how would we do that?
Maybe the inner part of the channel is polar, so it's reactive to the changes of the ions around it.
Okay, so we would want to build in parts of the protein which are charged, which have charged side chains. And these parts of the protein will be pushed or pulled by the electrical forces across the membrane. And that's exactly how it's designed. So I was drawing all these gates, but that's not actually what it looks like, right? The protein itself, the channel, actually has a lot of alpha helices which go through the membrane in kind of a cylindrical way. Okay, so lots of different alpha helices around a central pore. And every fourth of these alpha helices contains a lot of positively charged amino acids. What would those be, positively charged amino acids? No. Arginine, lysine, those are the main ones, and those are the ones that are actually in the channel. So every fourth of these helices will have a lot of arginine and lysine, which are positively charged. They're basic, and they're positively charged at physiological pH. Now, so the protein is in the membrane, and when we have resting potential, it's going to be very negative here and very positive here. So the positively charged amino acids and the domains, the alpha helices with these positively charged amino acids, are pulled inside towards the inside of the cell or inside of the membrane, and that keeps the channel closed. The central pore is closed. But once the outside becomes less positive and the inside becomes less negative, these alpha helices are being pulled out, and as they pull out, they rotate and they open the pore. Okay? And when the polarity changes again, they rotate back because they're pulled back into the cell and they close the pore. So this is the mechanism for the opening and closing. There's no real gate there, okay? But for the inactivation, that's a different thing. For the inactivation, so we have this thing that just closes and opens the pore based on how it's pulled or pushed outside of the cell. But then inside the cell, there is another domain of the protein which is quite unstructured. It's just kind of floating there on the inside. But as the pore opens, it really, really wants to kind of be sucked into the pore. okay? Because the domain it doesn't really want to be in solution. And as the pore opens, this is the perfect place for the domain to stick itself basically in. And this is the mechanism behind the inactivation. So the pore opens, and this thing is floating around, and once it finds the pore, it sticks itself in and blocks the pore. And that's the inactivation. And then you have to return the whole thing back to normal so that it pushes it out, the inactivation domain, and we can reuse the whole protein. So yeah, doesn't really look like this. Well, sort of does, okay? But this is how these voltage-gated ion channels work. And this is actually not just specific for the sodium channels, for the voltage-gated sodium channels. Basically, all the voltage-gated channels, calcium, potassium, etc., work the same way. It's the same mechanism behind them. They're actually even structurally very similar to each other. Okay, does it make sense how these things work? All right. Okay, we started at 1:45, right? I'm just aware of the time. All right. If there are no acute questions, let's take a very short break, 3 minutes or something, and we'll continue with the synapse. Okay. All right. Can we continue? All right, so as promised, we will now talk about the chemical synapse, so how it works that neurotransmitters are released into the synaptic cleft and things happen afterwards. But before we get there, let me just give you a couple more information about these voltage-gated sodium channels, which are, I think, more interesting and potentially clinically relevant. These voltage-gated sodium channels are absolutely essential for basically all animals that exist to actually survive, because all action potentials, all signaling in nervous systems, all muscle contraction, etc., in insects and marine mollusks and everywhere, require these voltage-gated sodium channels. They're absolutely crucial for the survival of basically everything, okay? Not plants, but all the animals. That is the reason why evolution, as you would expect, developed a lot of toxins that target specifically these voltage-gated sodium channels, because it's very easy to kill something by blocking these voltage-gated sodium channels, right? So there are many, many, many toxins in existence in nature which target these channels and either close them, block them, or open them without any control of the animal. Both of them will lead to paralysis and death, right? So it's a perfect way to kill something. I will give you two examples. One of them is something that you may have heard about. There is this fish in Japan called fugu, which is quite famous, and it's more famous. I don't think it's so famous for its taste, but it's famous because it contains this really, really powerful toxin, which is actually not produced by the fish. It's actually produced by some plankton that the fish eats and kind of stores this chemically very complex toxin. It's called tetrodotoxin. The name tetrodotoxin comes from the Latin name, well, kind of Greek name, for the fish, because it's called tetrodon. It has 4 teeth. It has more than 4 teeth, but that's where the name comes from. So it's called tetrodotoxin, but it comes from this fugu fish, actually from the plankton. Anyway, if you love chemistry, look up the structure. It's insane. It's really, really complex. Very cool. And tetrodotoxin is a very powerful blocker of these voltage-gated sodium channels. So it will actually move into the pore and block the pore. Okay, extremely effective. The lethal dose in humans is something like 500 micrograms or something like that. I had written somewhere, yeah, 500 micrograms if you inject it. So it's a tiny, tiny amount that will kill a human. Okay, it's very, very toxic. A different example. But this is really more for plant lovers. It's called aconitin. It's a plant toxin, very, very toxic, and it's actually from a plant that grows here in Czechia and Slovakia in Central Europe. Those of you who come from other parts of the world might not know it, or you have different species of this. It's called Aconitum napellus. It's a beautiful, beautiful blue flowering plant that sometimes people grow in their gardens. It's really, really nice. And it contains this aconitin, Yeah, you can look it up what it looks like. It's quite a nice thing. And the aconitin, which is one of the most toxic toxins that we have in our nature here, okay? So people who come from Australia, they have, you know, everything is toxic and everything will kill you. Here we don't have that many, but this one is one of them. And aconitin actually does the opposite from tetrodotoxin. It actually binds to these voltage-gated sodium channels and opens them and leaves them open, okay? The end result is the same. You get paralysis, okay? But the mechanism is completely different, okay? But as I said, there are hundreds, thousands of toxins which bind to these channels because it's such a good target for evolution and for this evolutionary strife. Actually this year, or the end of last year, a completely new class of medications to treat pain medications, non-opioid pain medications, was approved by FDA, which works by blocking these sodium channels. Okay, it's a completely new thing. I think it was this year, so it was like a couple months ago, and it's like, you know, after 50 years, a completely new class of pain medications which block these channels and block basically the action potentials coming from the pain stimuli. Super, super interesting. We'll see. I don't think it's registered in Europe yet, but it's an interesting thing. All right.
Can I ask you a question? Is it related to the Krebs cycle, to one of the enzymes that they're converting to aconitase, maybe?
Oh, the name aconitin. Yeah, so aconitase, the enzyme of the Krebs cycle, is called aconitase because its intermediate is called aconitic acid. And aconitic acid is also present in this aconitum plant, but it's not really related to the aconitin. I don't think it is. I don't think it's actually chemically related. But it comes from the same plant, yes. So the name, yeah, but it's a coincidence, really. Yeah. All right. Let's have a look at synapses. So this traditional chemical synapse is composed of two neurons, or bits of two neurons. One is the axon of the presynaptic neuron, and the other one is usually the dendrite of the postsynaptic neuron. We can also have synapses which go from axon to axon, so they're axo-axonal synapses. It's a possibility, but usually when we think about it, it's usually an axon to a dendrite, okay? Now, in the axonal ending of the presynaptic neuron, we get lots of different structures. We get cytoskeleton, we have microtubules there, actin cytoskeleton. We have loads of mitochondria there because we need a lot of ATP, okay? We have obviously huge numbers of ion channels in the membrane, etc. But what we don't have is the nucleus, ribosomes, and endoplasmic reticulum. So any proteins that we need to have here, they have to be produced in the cell body, which sometimes is very, very far away, and they have to be transported along the microtubules into the synapse. Again, this will become important towards the end of the lecture, right? So we can't synthesize proteins, but apart from that, we can synthesize pretty much everything that we need there, okay? So when we talk about neurotransmitters, all the neurotransmitters, with the exception of peptides and proteins, are synthesized directly in the axon, right? So the thing that makes the synapse the place of, you know, chemical signaling is that in the presynaptic neuron we have vesicles which contain a neurotransmitter, usually just one neurotransmitter, but yeah, there are exceptions, but usually it's just one neurotransmitter, one specific neurotransmitter, which is, when the action potential comes, which is released into the synaptic cleft, and this neurotransmitter then binds to some specific receptors and causes some stuff in the dendrite, right, or the axon, or wherever it's connected. Very basic structure, and I will want to spend most of the rest of the lecture talking about how this actually happens, so how these neurotransmitter vesicles are released, what is the molecular machinery behind this exocytosis. So that's going to be the topic for the rest of the lecture. But let me give you one other information, and that is that in recent years, well, maybe 40 years, so it's not that new, it's been found that this structure of a synapse for the central nervous system is too simplified, because we almost always have a projection of an astrocyte, of astroglia, which is in very intimate connection with the synapse. So this model where we have presynaptic neuron, postsynaptic neuron, and an astrocyte, you've seen the shape of astrocytes in some pictures. They have lots of projections, right? They're like little octopuses or something, right? If you haven't, look it up, okay? So it's a cell that has hundreds of different kind of projections to different neurons and to blood vessels and everywhere. So they're kind of connecting all the different bits. So imagine this is just one projection of the astrocyte, and this model is called a tripartite synapse. So synapse, which has 3 parts, right? Tripartite synapse. And it's a model which is now accepted as the usual thing in the central nervous system. So we always have 3 partners there, and the third one is astrocyte. And in the following lectures, we will see what these astrocytes can do. They can import and metabolize some neurotransmitters from the synapse, but they can also transmit some signals, okay? The astrocytes are actually connected by gap junctions to each other. So they form this massive network of astrocytes connected by gap junctions. So they can transmit some signals. They can't do action potentials. They will not give us this amazing self-amplified way of transmitting, but they can transmit some signals. They have neurotransmitter receptors on them, and it's clear that they play a super important role in signaling in the nervous system. We'll talk about them a little bit, probably not as much as they deserve, but that's how it is because we don't know all the details yet. So just think that there's always an astrocyte near a synapse which plays some important role. Right, let's now get to this exocytosis mechanism. So we have a vesicle, which is basically a membrane-bound part of the cell, which contains some neurotransmitter, okay, which was pumped in by some transporters, and this vesicle is the thing that we want to deliver to the membrane. So this would be the inside of the presynaptic neuron, right? We are here, right? Yeah? Okay. So this is the inside of the presynaptic neuron. This is the synaptic cleft. So we have this vesicle prepared, and we're just waiting for the action potential so that we can fuse this vesicle with the membrane and release the neurotransmitter into the synaptic cleft. How does this work? Well, first let me tell you that the mechanism that I will now show you for this exocytosis is actually the same for any kind of exocytosis. So it's not just specific to the synapse. If you have a gland which is releasing something from vesicles, the mechanism is the same. The mechanism is even the same between, for example, the endoplasmic reticulum and the Golgi apparatus. You know that there are some vesicles formed in the endoplasmic reticulum which then fuse with the Golgi. The mechanism I'll show you is basically the same there, okay? So whenever we have a vesicle which is fusing with the membrane, it's going to use this mechanism that I will just show you. So it's more general than just the synapse, right? But in the synapse, it happens as well. The key proteins which are responsible for this fusion of a vesicle with a membrane are called SNARE proteins. I'm writing in capitals because it's an acronym, so it's an abbreviation from very many weird words which you don't need to know, okay? They're called SNARE proteins. And these SNARE proteins have very special domains, bits of themselves, right? You know what a protein domain is, which are called SNARE domains. And what's so special about these SNARE domains? Well, once they get close to each other enough, they kind of snap together, okay? Like a Velcro. You all know what a Velcro is? Yeah, hopefully, okay, right? So the same way, basically, nothing happens, and then they just kind of snapshot, and they are very, very tightly bound to each other, okay? And that's basically the mechanism behind what happens here, but I'll give you a bit more details about that. So we have SNARE proteins with SNARE domains. Once you get them close enough, they snap together and pull things together because they really want to be kind of intertwined between each other. Now, these SNARE proteins are divided, there are many of them in our body, in our cells, and they are divided broadly, functionally, into 2 groups. They are divided into T-SNARE and V-SNARE proteins. Now this V snare stands for vesicle. So those are snares which are on the vesicle, right? So this would be a V snare protein. And this is my rendering of the snare domain, okay? So this would be a V snare protein. The T snare, the T stands for target. It's the target membrane. So the T snares are going to be on the target membrane, just waiting there to be connected with the other snare domains like a Velcro, right? So we have V snares and T snares. Now, I will give you the specific names of the V snares and T snares that are actually in the synapse, but be aware that there are many more of them. But the reason why I'm telling you what they're called, specifically in the synapse, is that, as we'll see towards the end of lecture, they are specific, once again, specific toxins causing specific diseases which attack directly these V snares and T snares, okay? So they are actually clinically relevant. That's where I'm telling you the names of them. So the V snare that I just drew here is called synaptobrevin. Synaptobrevin. Yeah, you know, weird names, but that's how it works. The T snare that I drew here in yellow is called syntaxin. Let me use the yellow. Syntaxin. And to complete our protein machinery, there's another T snare there, which is called SNAP25. I'm going to write it down. But this last snare protein, it's a T snare protein because it's associated with the target membrane, is a bit weird. These snare proteins are normal transmembrane proteins. You can see that they go through the membrane, and then there's the snare domain. And then they go through the membrane, and there's the snare domain. SNAP25 is a bit weird because it has two snare domains, and it is not a transmembrane protein. So it actually has two snare domains like this. And it doesn't go through the plasma membrane. It's only attached there by means of lipid anchors. You may have heard, and you will hear again, that there is a post-translational modification of proteins where we can add some lipids to the protein, which allows the protein to stick closely to a membrane. And it could be palmitic acid, as is the case here. So we can use fatty acid or myristic acid. Or it could be some isoprenoid like farnesyl. Now, you heard about small GTPases. No, not yet. Rus protein. That's a small GTPase, okay? Which is attached to the plasma membrane by means of farnesyl anchors, okay? So it's a common thing to use some lipid anchors to allow the protein to be attached to a membrane without having to go through the membrane like normal integral membrane proteins, okay? So SNAP25 is not a transmembrane protein. By means of these palmitic acid groups, it kind of sits on the membrane. Good. So now we have three different snare proteins, okay? Synaptobrevin, syntaxin, and SNAP25. And they are just waiting there to combine their snare proteins and to pull the vesicle containing the neurotransmitter towards the membrane. And for that, we need some signal. We need some signal that tells it, now pull everything together. And the signal is provided by calcium, as is often the case, right? So as the action potential comes along towards the end of the axon, in addition to voltage-gated sodium channels, we also have voltage-gated calcium channels, which also open, and they allow calcium to go in. In fact, the voltage-gated calcium channels are very close to this whole exocytic machinery. Remember, in the cells, when something needs to work together, we always want it to be very, very close to each other, right? We don't really want to have things diffusing very far. So here, the calcium channels are basically very close to the vesicles. Calcium goes in and binds to a calcium sensor, which is on the vesicle. So this is not a SNARE protein, this is just a calcium sensor. And this calcium sensor is called synaptotagmin. Synaptotagmin. And it's a protein which binds calcium, and as it binds calcium, it changes its conformation. And the changed protein has a very high affinity to phospholipids. So when there's no calcium, it just sits there. But when the calcium comes in, it kind of exposes a new part of the protein, which has a very high affinity to phospholipids. And what happens is that the synaptotagmin gets pulled towards the plasma membrane because there are phospholipids there, and it has a high affinity for phospholipids. It pulls the vesicle with it, and as these SNARE domains come together, they snap together, okay, as a Velcro, and that ends the pulling of the vesicle towards the membrane. So kind of the intermediate state is going to look like this, where all the SNARE domains are going to be combined together in a very tight kind of Velcro-like structure. And as the membranes of the vesicle and the plasma membrane, as the lipids of the vesicle and the plasma membrane get together, they fuse together and allow the neurotransmitter to exit the vesicle. Okay? Once again, this is a general method for exocytosis. The names of the proteins might be different, but the general method is the same basically in any tissue that does this. Okay?
Why is, maybe I missed something, but why is SNAP25 important for this?
Yeah, it's just there. We need more than 2 proteins. We need a third one there. Okay, whatever. I mean, to be completely honest, I'm leaving out about 60 or 80 other proteins which are part of this. It's way more complicated. There are a lot of regulatory proteins, there are a lot of chaperones that keep things, etc., etc. So there are definitely reasons for having, 4 of them there, because there are many, many other proteins involved in that. So I'm kind of pulling out the most important ones, or the ones that make sense altogether, but there are many more proteins. So there is definitely some good reasons for having it there. Maybe it is for the power that we need to produce in order to fuse the membranes. Maybe it's for some regulatory reasons. Yeah. But as I say, I'm leaving out the vast majority of proteins that are involved in there. There was another. No. Yep. Okay. Well, we will discuss it a little bit more in later lectures, okay? There are many functions of the astrocyte. One of them is that they can import the neurotransmitter from the synapse and then process it and do something with it. That's something that we'll see in the next lecture, okay? But they also react to neurotransmitters. For example, one of the things, and I'll leave you at that because well, we do have a bit of time. For example, astrocytes are responsible for getting enough glucose, enough energy to all the neurons, because they are attached to blood vessels, okay? And they can import glucose, turn it to lactate, and give it to neurons. Now, one of the things that happen is that as the neurons are producing neurotransmitters, which means that they are active and they need a lot of energy, the astrocyte has receptors to find out, oh, a lot of neurotransmitters being produced, and it means that it starts sucking more glucose from the blood vessel and providing it to the neuron because it knows that the neuron needs it, okay? So there are a lot of functions of the astrocyte which are related to synaptic transmission. Without it wouldn't work. But we will get to some of them in later lectures, okay? Any questions about the exocytotic machinery? Yes?
So we have all of these proteins in almost every cell.
Yes. Yes, we do. I mean, the specific names of the proteins might be different, okay? Because as you can see, these are all called synapto something. So these are often associated specifically with synapses, but we will have very closely related proteins in other cells as well. Yep, absolutely. Good. So the last few things that I want to talk about with respect to exocytosis is the same way the voltage-gated sodium channels are such a crucial thing that, you know, it pays evolution to develop toxins. The exocytotic machinery is clearly as important, because without it, we don't get any neurotransmission at all, okay? So we do have also toxins which interfere with this machinery. But these toxins are a little bit more complicated. So I told you to look up the structure of tetrodotoxin, and you will see that it's a complicated structure. But here, these toxins that influence the exocytosis are actually much bigger. They are proteins. They are proteins, and many of them are even enzymes. And they are produced by bacteria. So they are bacterial toxins, and they cause diseases that you will, well, hopefully you won't come across them, but you may. And that's why I want to tell you about the mechanism of action, because you will hear about these bacteria later on, and you've heard about these diseases, and as you'll see, one of these toxins is even used in medicine. So, how these toxins work. They are, both of the toxins that I'll be talking about, are produced by bacteria from the genus Clostridium. Clostridium, you will learn about them in microbiology. And they are a genus of anaerobic bacteria. These bacteria really don't like oxygen. They love to live in places when there's no oxygen, okay? Some people have them in their guts because there's very little oxygen there, etc. So there are many different Clostridium bacteria. You will hear a lot about them in microbiology because they're causing a lot of diseases, etc. Now, the first Clostridium bacterium, which is producing the first toxin I want to talk about, is called Clostridium botulinum. And it produces botulotoxin. Now, have you heard about botulism? What it is? What kind of disease it is and how it's caused? Huh? Correct. So usually it is a disease, poisoning really, caused by canned food. Because when you can food, usually there's not a lot of oxygen. And if you don't can it properly, if you don't heat it up properly, often it's with meat and stuff like that. If you don't heat it up properly, there are perfect conditions for these Clostridium bacteria to grow there. And they grow, and they produce these botulotoxins. There is emphasis on the plural, because there's more than one. And once you open the can and you start eating it, basically it's not the bacteria that will poison you. It's also a possibility, but usually that's not the case. But it's the toxins. And these toxins are fairly thermostable. So even if you heat it up, or even if you cook it, a lot of the toxins can actually survive the cooking, even though they're proteins, but they're quite stable, okay? So you eat these toxins, and these toxins get into your body. Now, the super interesting thing about these toxins is how clever they are, how clever evolution developed them. Because the main component of the toxin needs to get into the nerve cell. It needs to cross the membrane. And remember, it's a big protein. It's not easy for a big protein to cross the membrane. So it needs to get specifically into the nerve endings. And there, the main component acts as a protease. It's a protein degrade, very specific protease, which in the case of botulotoxin breaks down SNAP25. So we will come into the cell, find SNAP25, and snap it in half, or however many pieces. Just destroy it. Just this one protein, that's it. It doesn't do anything else, okay? Now, the clever thing about these toxins is that you can't get a big protein into a cell. It's impossible. You can't cross the membrane. So actually, the botulotoxins have several components. One of them will find the nerve cell and bind to a receptor and kind of make a docking station there. Another component will come in and make a pore, make a hole into the membrane. And the third component comes in and goes through the pore and actually destroys SNAP25. So these bacteria have developed really, really advanced kind of biological warfare, really, where they have different components which target these toxins specifically when they are needed, and they are extremely specific. They will really just cut this one protein, and that's it, okay? But of course, without SNAP25, there is no exocytosis, okay? You don't get any synaptic transmission, and the result is paralysis. So this botulism disease or poisoning from this canned food, it doesn't have to be canned food, but that's the typical example, is that people may have difficulties breathing, okay? They may have difficulties with autonomous functions because there's basically ongoing paralysis, and in the end, they can stop breathing or they can even die because of this poisoning, okay? Now, these toxins are some of the most toxic substances ever known, okay? They are in fractions of micrograms, in nanograms, they can kill you, okay? They are insanely potent, okay? Now, you may have heard, well, most of you probably have heard about Botox, right? Something used in cosmetics, Botox injections. Probably a lot of people have heard about it. Well, that's botulotoxin, okay? That's what they use. What it's used for, Botox?
Muscle relaxant.
Correct, it's a muscle relaxant, even though that's not how they market it, but that's exactly what it is, right? So basically they paralyze your facial muscles, and the gravity pulls down the skin and you look younger, but obviously you can't use your facial muscles, so, and you've probably seen videos of people who had too much Botox where they can't smile because, you know, their facial muscles are paralyzed. Why do we use botulotoxin for that? Well, we could use some other muscle relaxant, right? But the problem is that most muscle relaxants are very short acting, so you'd have to be like giving these injections all the time and, you know, it'd be a problem. But with botulotoxin, once you destroy the SNAP25, it takes a very long time for the nerve cell to produce new SNAP25 in the nucleus and then transport it all the way into the synapse, right? So you give the injection close to the synapses and you can wait months. The effect usually takes months because it takes months for the cell to produce new SNAP25 and actually deliver it where it's needed. So it's very, you know, it's very practical to use botulotoxin rather than something else. So this is the mechanism behind Botox. You destroy your SNAP25 and you paralyze your facial muscles.
If so, how we can recover someone which is affected by the toxin?
Yeah, so the first thing that you do is you give antibodies, you give antitoxin to bind any toxin which has not gone into the nerve cells, and that's the main treatment. The rest of it, once they, for example, can't breathe or something, you just have to do supporting therapy. So you have to ventilate them and just wait for everything to get back to normal. I mean, most poisonings are not that bad that people would actually, I mean, you know, in the past 70 years or something, because there's a lot of control, how canned food is made, etc. So it's very unusual. But yes, if you had a patient like that, first you give antitoxin, and then just supporting measures and supporting treatments. Yeah, there's no specific treatment. You can't give them more SNAP25. That can't be done. Now, the other quite closely related toxin, which is also produced by a bacterium from the genus Clostridium, but this time it's Clostridium tetani, which produces tetanotoxin, or once again tetanotoxins, because there are more components. It actually works in a very similar way to how botulotoxin works. And tetanotoxin breaks down synaptobrevin. Okay, so botulotoxin breaks down SNAP25, the other one synaptobrevin, but basically the result is the same. You don't get any neurotransmission, right? Now, those of you who have heard about tetanus, the disease which is caused by Clostridium tetani, might think, well, that's a bit weird because botulism causes paralysis. But tetanus has significantly different symptoms. What are they? Spasms, exactly. So people with tetanus will have actually extreme spasms of the muscles, oftentimes elicited by like a sound, or somebody, you know, claps their hands and they go into whole body spasms, okay? So it looks like it's doing the opposite, okay? Well, it sort of is doing the opposite and sort of isn't. The reason why we get so very different results from these kind of same mechanism inside the cell is that these two toxins are targeted to different neurons. So they don't just go to any neuron, but they are targeted to specific neurons. And why botulotoxin goes specifically to motor neurons, neurons that affect directly the movement of muscles, tetanotoxin is directly targeted to inhibitory neurons in the central nervous system. So it blocks the transmission of neurons which normally inhibit the function, they try to prevent overaction of the nervous system. But if you block the neurotransmission there, you get this overactivity, which is how you get those spasms by sound or something, and the brain just overreacts and causes these massive spasms. So the molecular mechanism is the same, but the end result is massively different because they are targeted to different types of neurons. All right, that's it. Questions? Yes?
Also because it's like one is a T-SNARE, one is a T-SNARE.
No, not really. That's not the reason that one is a T-SNARE. We need both of them, or all three of them, in order to get exocytosis. So it's really about not the protease component. It's about the component that kind of docks to the neuron, and that's specific to some types of neurons, and the other one is specific to other types of neurons. So it's not really about the protease itself, but it's about how it gets into the cell. Any other questions? No? All right. I'll see you tomorrow, and we'll talk about neurotransmitters tomorrow.